going to finish Chapter 9. We're going to look at Part B of Chapter 9. Part B is going to look at whole muscle contraction, so we'll be looking at the gross anatomy or physiology of muscle, or muscle contracting as a whole. So a couple of principles are going to be the same. We'll still be talking about excitation, contraction, coupling. We'll also be talking about cross-bridge cycling. But we want to talk about them and how they produce muscle tension. So muscle tension is going to be the force exerted on any resistance or load that a muscle is trying to overcome in order to move that object or load. Different types of contraction. Some may cause muscle shortening or lengthening, others may not. We have isometric contraction and isotonic contraction. Isometric contractions are going to occur when muscles do not change length during a contraction, such as when you're holding a book in midair. Those muscle fibers that are used to produce a force have created tension, but no motion is occurring, so no cross-bridge cycling is actually causing shortening of sarcomeres during isometric contraction. During an isotonic contraction, we're going to see shortening of the muscle. We'll see that muscle tension be stronger than the load or resistance that muscle is trying to overcome. And the muscle shortening will occur when the sarcomeres collectively in a muscle fiber and then collectively in the muscle itself all shorten. So during isotonic contractions, muscles change length. And picking up a book is an example of an isotonic contraction. So isometric contractions, no shortening or no length changes for the muscle. Isotonic contraction, we will have length changes. Usually it's going to be muscle shortening, but it could be muscle lengthening. We also want to talk about motor units. Motor units are a nerve muscle functional unit. They're made up between the many axons and their axon terminals from a motor nerve and the sarcolemmas of the muscle fibers in a muscle. So that's what we're looking at here. Is basically a bigger version of the neuromuscular junction. Because a motor nerve contains many axons, not just one, and all those axons have branching axon terminals that get really close to their associated muscle fibers. So motor neurons plus all of the skeletal muscle fibers that they innervate, that's what is considered a motor unit. So here's the proper definition that we're given in your lecture textbook and this lecture PowerPoint, And motor units consist of a motor neuron and again, all the skeletal muscle fibers that it supplies. We can have smaller or bigger muscle units. The largest motor units contain or innervate several hundred skeletal muscle fibers. And the smaller the motor unit, the more it's associated with greater or fine control. Here are two examples of motor units. We have one in purple and one in red. The one in purple innervates 2 skeletal muscle fibers, and the one in red innervates 3 skeletal muscle fibers. Both of these axons, or motor neurons, are coming from a group of axons called a spinal nerve. So just like muscle is a group of fascicles, which is a group of skeletal muscle fiber, a spinal nerve is going to be a group of axons that are also arranged in similar structures like fascicles. A muscle twitch is going to be a really simple contraction. So a single action potential going from a motor neuron to a muscle fiber, that would be an example of a muscle twitch. You can measure a muscle twitch and other muscle activity via a myogram. A muscle twitch is going to be the simplest type of contraction because it's going to be just one signal that's stimulating the muscle fiber, and we'll see not only the contraction or tension rise in that skeletal muscle, we'll see it relax as well. There are 3 phases to a muscle twitch: a latent period, a period of contraction, a period of relaxation. Those are the 3. During the latent period, we're not going to see any muscle tension because during the latent period, we are not going to see any cross-bridge formation. Cross-bridge formation is what creates or produces tension, whereas during the latent period, we are still looking at or seeing a muscle that's going through the excitation, contraction, coupling phase, where we're trying to link an action potential being generated in the sarcolemma of a skeletal muscle fiber to not only calcium release from the sarcoplasmic reticulum, but then ATP use by the myosin heads of the thick filament. So during the latent period, we are not going to see any tension. But during the period of contraction, we have cross-bridge formation, so tension increases, and then we'll have the period of relaxation. When we get rid of that calcium, that is a final go signal for contraction, that calcium will be actively pumped back into the sarcoplasmic reticulum. And when it's no longer in the sarcoplasm, we'll see that tension decline all the way to zero. So 3 phases to a muscle twitch. We have the period of time in which we are getting to the cross-bridge formation by linking excitation or the action potential to the contraction phase of skeletal muscle. And then we have the actual contraction, and then its relaxation. We'll also see that muscle contracts a lot faster than it relaxes. It takes about only 30 seconds for us to reach the peak tension of this muscle twitch. But it takes 110 seconds for the relaxation of this muscle twitch. And depending on the size of a muscle or the enzymes or the amount of nutrients reserved in different muscles, muscles will contract and relax at different rates. Here are 3 examples. We can see that the smaller lateral rectus muscle is able to contract and relax quicker than the larger gastrocnemius and soleus muscles. Next, we're going to look at multiple stimuli that cause more than just a single muscle twitch. They instead cause graded muscle responses. So these are more normal muscle contractions that are going to be pretty smooth. They're going to be useful for moving objects or moving parts of the body. So a muscle twitch is not normal. It's not going to be seen really anywhere other than a lab setting or with neuromuscular problems. Not to say if you've had an eye twitch or a random muscle twitch that you have neuromuscular problems, but it is a momentary imbalance in ions and other components of muscle contraction that cause these individual muscle twitches. So graded muscle responses are going to vary a couple of things. They can vary the strength of contraction. Basically, they can vary how many motor units are going to be used to stimulate that muscle. Or they can vary the frequency of stimulation, meaning they can increase how fast action potentials reach that skeletal muscle fiber, or they can decrease it. Increasing the rate of stimulation or the frequency of stimulation is going to produce the graded muscle responses. Same thing for increasing the strength of stimulation is going to produce those graded muscle responses. So a single stimulus is going to be a muscle twitch. Multiple stimuli can produce what's called wave summation or temporal summation. So let's look at the muscle twitch first so we can compare it later to the temporal or wave summation, which is a type of graded muscle response. So here we have a stimulus that causes a muscle twitch, and we see both the contraction and relaxation phases. And then it's a couple of milliseconds before we get another stimulus that causes a separate muscle twitch, and we're able to see contraction and relaxation. No summation is seen here because the stimuli that occur that create the 2 muscle twitches are too far apart. If we were to increase the frequency of the stimuli, meaning the stimuli occurred closer, then the tension built up by these separate muscle twitches would actually be added together, and we would get tension that is higher than this maximal tension for a single twitch. But if it's higher than the maximal tension for a single twitch, we're not going to be calling it a twitch. It'll be a wave summation or graded muscle response. So wave or temporal summation would occur if you had consecutive stimuli applied during the relaxation period. If it was applied during the relaxation period of each preceding muscle contraction, then you would start to see that each subsequent contraction was builds upon that previous contraction so much that it's going to increase tension before the relaxation period ends. So wave summation is the same thing as temporal summation. And because the muscle fibers don't have time to completely relax before another stimulus arrives, we see that the tension buildup from each contraction gets greater and greater as we get more stimuli. And that's because we have additional calcium being pumped into the sarcoplasm and acting as that go signal for contraction and prolonging the cross-bridge cycle. So here we can see temporal or wave summation. the stimuli are much closer together than the example we saw with the individual muscle twitches. So before each contraction is able to relax, we're going to have another contraction, and that's going to build off of or piggyback on the previous tension that wasn't yet dissipated. So here we see unfused or incomplete tetanus, where we're still seeing partial relaxation between each stimulus. So again, the term I just used was unfused or incomplete tetanus, and that's being caused by an increase in frequency of stimuli. So if stimuli frequency increases, muscle tension reaches near maximum, and even this unfused or incomplete tetanus can be a smooth, continuous contraction because of that temporal wave summation that occurs. So unfused tetanus can also look like this, where we have even closer stimuli, and so we have even less relaxation between each contraction and relaxation phase. There is also fused or complete tetanus, and that would occur when the frequency of stimuli increased still further, so more than unfused or incomplete tetanus. And in this case, with complete tetanus, you would see that individual muscle contractions completely fuse to produce one large muscle contraction. So we'll see a sustained contraction plateau, and we won't see that relaxation in between each stimulus. Now, if this were to go on too long, we could see that muscle fatigue resulted. So is our fused tetanus. Here's our plateau. We see the stimuli are closer than ever. And if this were to be sustained, such as when someone gets a surge of adrenaline and can lift a car in extreme rare circumstances, then we would see muscle fatigue result. We also want to talk about the other way that we can increase the contraction of muscle, or that we can cause a graded muscle response, and that was to increase stimulus strength, or to use more motor units. So this could be called multiple motor unit summation or motor unit recruitment. And there's a couple of different types of stimuli involved in recruitment. If only a couple of motor units are used, their stimuli may be subthreshold, meaning not enough skeletal muscle cells in that muscle are going to contract in order to cause whole muscle contraction. There's also a threshold stimulus where you have a little bit more or more skeletal muscle fibers able to contract, so you see some shortening or maybe some lengthening of that skeletal muscle fiber, but it's not a maximal contraction. We're not saying maximum contractile force, which only occurs with a maximal stimulus, meaning that all motor units have been recruited for that muscle as an organ, and we're going to see the strongest possible contraction from that muscle. That's what we see in these charts. We're looking at motor unit recruitment. A subthreshold stimulus is not going to cause muscle shortening. Very few skeletal muscle fibers, if any, will be contracted. And then with a threshold stimulus, we'll have a couple more skeletal muscle fibers contracted. You may see a weak muscle contraction during a threshold stimulus. With a maximum threshold stimulus, we will see all motor units recruited, therefore all skeletal muscle fibers innervated by those motor units contracted. We'll see the maximum contractile force for that organ. The recruitment process does also work on a size principle, which is really simple. So motor units that are the smallest are going to be recruited first, then slightly larger motor units, and then finally the largest motor units. So only when the most powerful contractions are needed would the largest motor units be activated. That's what this image shows. We start with the smallest motor units. So smaller motor units innervate fewer skeletal muscle fibers. Medium fiber motor units innervate a couple more skeletal muscle fibers. And the largest motor units innervate thousands of skeletal muscle fibers. Next, we're going to look at muscle tone. Muscle tone is a slightly contracted state. that all muscles exist in. It's constant. It's going to be due to spinal reflexes. And in any relaxed skeletal muscle, there's going to be a small number of contractions that continuously occur. Again, muscle tone is constant. And this will create or could be observed as firmness in a muscle. And it causes these contractions that we call muscle tone. Muscle tone is able to maintain body posture. Also, it can increase muscle readiness or its readiness to respond, to contract. It also keeps muscles healthy. We're going to look just quickly at the terms we started this chapter on, which are isotonic and isometric contractions. We want to see examples in both images and talk about different types of isotonic contractions. So isotonic contractions were contractions that caused changes in muscle length. And most commonly, this is going to be a concentric isotonic contraction. Concentric isotonic contractions occur when muscles shorten as they do work or move a load. So your bicep picking up a book or contracting and shortening to pick up that book is a great example of a concentric isotonic contraction. Whereas an eccentric contraction is when the muscle doesn't shorten necessarily, but it does change length. The muscle is going to lengthen during an eccentric isotonic contraction. It still generates force, and laying a book down, causing the bicep to lengthen while you're doing that, is another example of an eccentric isotonic contraction. Another example would be contraction of your gastrocnemius muscle. When you walk or perform an ambulatory movement, your gastrocnemius will lengthen. So it's generating force by lengthening during an ambulatory step. Make sure you can recognize an illustration of isotonic and isometric contractions. So this is the one usually used for isotonic contractions, but it is showing isotonic concentric contractions, because what we see is a muscle at rest, and then when tension builds up, it's able to change the length of the muscle by shortening it. So this change in muscle length is an isotonic contraction. So we see that resting length is going to experience a decrease as tension builds up. Again, with an isometric contraction, we see no shortening or lengthening of the muscle. This would be like trying to push over a wall or just pick up something that's way too heavy to pick up with your skeletal muscles. So the load's going to be greater than the maximum tension that the skeletal muscles can perform with their cross-bridge cycling. Don't forget that the electrochemical mechanical events that we talked about microscopically are still occurring, and that's why isotonic and isometric contractions occur. During an isotonic contraction, cross-bridge cycling is going to be productive. The myosin heads are going to be able to move along the actin or thin filaments and cause sarcomere shortening. Whereas during isometric contractions, myosin heads are just going to spin their wheels or not be able to get a good hold on those actin binding sites or those thin filaments. Here's a good diagram for isometric contractions. We see resting length does not change during tension buildup in an isometric contraction. Tension does build up in both isotonic and isometric contractions, but only in isotonic contractions is there enough tension to overcome the load trying to be moved. Here we can see for an isometric contraction that tension builds, but we have no change in length for the muscle. Next, we're going to talk about how are we creating the energy that is used for muscle contraction. How are we creating the energy that is used to pump calcium out of the sarcoplasm and back to the sarcoplasmic reticulum? How are we creating ATP to create an end-plate potential or to open sodium-potassium channels during excitation, contraction coupling? And specifically, how are we creating ATP, which is going to power the cross-bridge cycle? It is true that skeletal muscle does store a little bit of ATP, only about 4 to 6 seconds worth. And this ATP is a great source of energy because we can break it down into ADP, and that breakdown process produces energy. But since we only store 4 to 6 seconds worth in skeletal muscle, there has to be other ways to create ATP. Some that are going to be quick, just like the energy stores are because they're readily available to use, and others that are going to be able to sustain the energy production needed, but take a lot longer to produce that ATP. So here are 3 mechanisms for producing ATP in skeletal muscle. We have direct phosphorylation of ADP by creatine phosphate. This process is quick and it's simple. We're going to take a phosphate off of creatine phosphate and add it to ADP, which produces ATP. For the anaerobic pathway, we're going to see that a process breaks down glucose and produces lactic acid. Anaerobic means a process that occurs without the use of oxygen. Glycolysis is always an anaerobic process. It does not require oxygen. However, if there is oxygen present, the products of glycolysis can be used in the aerobic pathway. The aerobic pathway uses the products of glycolysis to create a lot more ATP than either direct phosphorylation of ADP by creatine phosphate or the anaerobic pathway. But unlike our first and second steps, which are quick, the aerobic pathway takes hours. So let's look at these processes in more detail. Direct phosphorylation of ADP by creatine phosphate, we're going to see an enzyme called creatine kinase be used to take a phosphate from creatine phosphate and add it to ADP to make ATP. It's quick, it only takes 15 seconds, and we get our energy that we wanted, or we get 1 ATP for every creatine phosphate we use. Here's a good illustration to think or use to think about direct phosphorylation. We take phosphate from creatine phosphate, add it to ADP, we get our energy, and a byproduct is creatine. Very quick, but it only gives us 1 ATP molecule for an entire chemical process. The anaerobic pathway does not use oxygen. It doesn't require oxygen. And we're going to break down sugar, glucose, into pyruvic acid. Without oxygen present, pyruvic acid is going to be broken down into lactic acid. And lactic acid, if it's not pumped to organs, can build up in muscle and make your muscle sore. You get 2 ATPs for every glucose we break down. The word glycolysis means sugar breakdown. Glyco for sugar, lysis for breakdown. So normally, if there was oxygen present, pyruvic acid would go into a mitochondria and be used in the aerobic pathway to make a lot of ATP. That's the process that takes hours. But without oxygen present, we're going to use the pyruvic acid and convert it to lactic acid, which can be recycled in other organs and used again in glycolysis. Lactic acid should diffuse into the bloodstream. It can be used by other organs like the liver, kidneys, and the heart. In those organs and in skeletal muscle, it can be converted from lactic acid back to pyruvic acid or even glucose and then reused for the process of anaerobic glycolysis. This is a really quick process, but it takes a little longer than 15 seconds. It takes about 30 to 40 seconds, but we do get 2 ATP instead of 1 ATP like we got in direct phosphorylation. So we get 2 ATP for every glucose we break down. This process does not use oxygen. Pyruvic acid is the normal product, but without oxygen, pyruvic acid gets converted to lactic acid, which can be recycled to do this process all over again. And this process takes 30 to 40 seconds. And then last but not least, for our energy producing mechanisms is the aerobic respiratory mechanism. The aerobic pathway uses oxygen, and this is what most skeletal muscle cells use to produce most of their ATP. We can see that it produces 95% of ATP during rest or light to moderate activity. It's a lot slower than the other pathways. The anaerobic pathway took 30 to 40 seconds. This is going to take hours. We're going to use mitochondria and oxygen to break down glucose. But by using mitochondria, we're able to make more than just 2 ATP. We can make 32 ATP per glucose. We can make 30 more ATP from 1 glucose molecule than the anaerobic pathway could. We can also use amino acids and fatty acids as a nutrient molecule for this process. We don't have to use glucose, even though glucose is the one that is most readily used. So we can use glucose, fatty acids, or amino acids when oxygen are present to perform aerobic respiration in a mitochondria. This process produces up to 32 ATP to power skeletal muscle contraction and the processes therein. So even though oxygen is required, we get a lot of bang for our buck, but it's going to take hours. So a couple of energy systems for using these three mechanisms. One is going to be aerobic endurance. So we see the use of aerobic respiration during aerobic endurance. This includes that light to moderate activity. And that's why you can continue for hours, because as long as you have that ATP, it's going to be consistent, it's going to be more than the other two processes, and the length of time that muscle contracts can be longer during aerobic endurance. Anaerobic threshold is going to be the point at which muscle metabolism converts to anaerobic pathways. So during quick, high-intensity exercises, your body does not have a chance to use aerobic endurance and will instead use anaerobic threshold mechanisms. So during short duration or high-intensity exercise, we're going to use our ATP stores. We're going to use direct phosphorylation of ADP with creatine phosphate because we just want to create ATP as quick as possible for our short duration but high-intensity exercise. We want to have quick energy for a short period of time, which is great for creatine phosphate and the anaerobic pathway. But any sustained, prolonged duration exercise must use the aerobic mechanism because even though it takes hours, it's also able to sustain hours of exercise. And then one of the last things we're going to look at is muscle fatigue. And muscle fatigue is when muscle is unable to contract. Usually it's because of an ionic imbalance. Potassium, sodium, calcium are very important to muscles and how they contract. It's involved in both that depolarization, repolarization, and calcium's involved in the excitation-contraction coupling step. So if something's wrong with the ions, muscles can become fatigued and unable to contract. Very rarely is it going to be a lack of ATP that causes muscle fatigue, but in extreme circumstances, a lack of ATP could be the reason that muscles cannot contract. So muscle fatigue is when muscle fibers stop contraction. Again, it could be when inadequate amounts of ATP are available, but normally it's because of ionic imbalances. Sometimes it's because of a lack of oxygen or glycogen or maybe the accumulation of lactic acid. Lactic acid is a byproduct from ATP production that we saw with the anaerobic glycolysis mechanism. And this together with the lack of ATP could all contribute to muscle fatigue. What we call excess post-exercise oxygen consumption, a mouthful, but what this is what a muscle has to do to contract again when it's stimulated. Because during muscle fatigue, even if a muscle is stimulated, it's not going to contract. It's not going to contract until it has oxygen again, till it gets rid of excess lactic acid, till it builds up its glycogen stores, until it regenerates ATP stores and creatine phosphate. So what a muscle has to do, all these replenishing steps, are called the excess post-exercise oxygen consumption, or EPOC, EPIC. I much prefer the earlier or former term for this, which is oxygen debt, because it mentions one of the things that we have to replenish for fatigue to go by the wayside. So muscle fatigue is when muscles can't contract, even when stimulated. The oxygen debt, or excess post-exercise oxygen consumption, is the debt that muscle must pay when it's fatigued in order to once again be able to contract during that stimulation process. And that is it for Part B of Chapter 9. If you have any questions, please feel free to email me or send me a message. Make sure that you go back and look at Part A and look at the videos that go over the neuromuscular junction, excitation, contraction, coupling, and cross-bridge cycling. Understanding the anatomy microscopically and grossly for muscle is really going to help understanding the physiology both microscopically and on an organ level. And that's it for Chapter 9. I hope this was informative.